And today we will start the program with Björn Schumacher, Professor Björn Schumacher. Again, a shameless promotion of his book from my side, The Mystery of Human Aging, Surprising Insights from the Science That Is Still Young. Beautiful title. Please find it online, find it at Amazon and read it. Professor Schumacher is a full professor and the director of the Institute for Genome Stability in Aging and Disease at the University of Cologne. He holds many other titles, and he is also, since 2014, he is the president of the German Society for Aging Research. He focuses on C. elegans, but also focuses broadly on the molecular mechanisms through which DNA damage contributes to cancer development and age-associated diseases and pathologies. So, Dr. Schumacher, off to you.
Thank you so much, Alex, for the kind introduction. And Alex and Morten and all your staff for organizing this wonderful meeting. Very exciting. So I'm going to talk to you about genome stability mechanisms in aging. And let me just go to the pointer. So what's important to realize is what actually comes from August Weismann's lab in the late 19th century, when he proposed that germ cells and only germ cells immortally perpetuate the genetic information. He realized that immortality of the soma would be completely futile in terms of contributing to evolutionary fitness. Therefore, the soma can decay, age, and die as soon as the genes have been safely transferred to the next generation. Based on these concepts, Tom Kirkwood proposed the disposable soma theory of aging, which pertains that the soma needs to be maintained to allow successful reproduction upon which the soma can be disposed of. And based on these considerations, there are three important questions that are really at the heart of our interest in aging. First, how is somatic maintenance adapted to the requirements of the germline? If you imagine a human were to die within the first decade of life, there would be no reproduction and extinction of species. Secondly, how do germ cells actually manage to be immortal? And third, which are the processes that determine the maintenance of the soma and ultimately control aging? Now, DNA damage is really at the core of the fundamental underlying mechanisms of driving the aging process. DNA damage occurs in all our cells, in all our genomes by the tens of thousands every day. And damage can contribute to the functional decline and therefore promotes aging. DNA damage also gives rise to all sorts of mutations that can then transform cells into cancer cells. Now, if you think about recent understanding of the consequences of DNA damage, it really mechanistically is connected to all these hallmarks of aging that we have heard earlier in this conference. Without going into more detail there, I want to just walk you through some of the discoveries that led us to better understand these three components we're interested in. So some time ago, Maria Amoleva, when she was in the lab, asked whether how we can identify somatic adaptations to the requirements of the germline. And in C. elegans, we have a fabulous system for investigating what would happen when the germline, here as a schematic representation, carries DNA damage. And in a nutshell, what we found was actually that DNA damage in germ cells triggers an ERK-MAP kinase-induced innate immune response that in the soma then activates the ubiquitin proteasome, and the enhanced proteostasis confers stress resistance and somatic endurance, which we postulate then allows germ cells to repair their genomes and resume offspring generation later in life. Thus, it's the mechanisms of extending reproductive lifespan. Now, secondly, how can you understand how germ cells manage to be immortal? And one prerequisite for the immortality is certainly the genome stability in primordial germ cells. Studying them in humans is very complicated, such as and even in mammals in general. Worms are a fabulous model because in the L1 larvae, there are only 2 primordial germ cells that when they encounter DNA damage, they will not divide, in contrast to wild-type animals that divide and can then form a germline. DNA repair deficiency, global genome nucleotide excision repair deficiency leads to their permanent arrest, and when the worms grow up to adulthood, they don't have a germ. Bourlignon in the lab asked how this works. And in a nutshell, I just give you the result because this has been recently published. When primordial germ cells experience DNA damage, they induce the P53, the elegans P53 that's called CEP1 that orchestrates the arrest of primordial germ cells. But this is regulated not cell autonomously, but by the somatic niche cells through FGF signaling. FGF is called AGL17, AGL15, the receptor of FGF, through ERGMAP kinase signaling, a translation initiator EFA4, a very specific regulator of very specific mRNA targets, including the FGF receptor, induces then its translation in a positive feedback, forward feedback. And again, FGF signaling through a distinct type of receptor, the SERF receptor tyrosine kinases, then triggers the P53 response. So it's so the soma controls the genome stability response in primordial germ cells. And we found that this is highly conserved and similarly present in the hair follicle stem cells, for example, which is an experimentally very accessible model in mammals. So what I want to talk to you about in more detail are these processes that determine somatic maintenance. And one of those processes is the maintenance of the chromatin structure, where histones have many distinct roles in chromatin condensation, DNA replication, transcription, and DNA repair, which of course for us is the major interest. Through a genetic screen, we found components of the MLL compass complex that mediate the mono, di, and trimethylation of the histone 3 lysine 4. And what we find there, when we look at UV irradiated animals that were treated at L1, then 2 days later, we assess their development through the L1, 2, 3, 4 larval stages. We find that here, for example, when 50% of the wild types are arrested, mutants in the MLL compass complex here are significantly more sensitive, while in contrast, the SBR5, the demethylase that removes di and monomethylation, has a significant resistance, so the opposite phenotype. That caught our interest, and it already here, because of the combination of the methyltransferase cofactors and the methylase, this already pointed to a role of dimethylation. We further explored that here, where we do the same experiment and see that mutants in SET17 is to methyltransferase for dimethylation, and it is highly sensitive, while the demethylase AMX1, specific for dimethylation, confers resistance, again pointing to dimethylation. We then looked at DNA damage-driven aging. Upon UV treatment, lifespan is curtailed, and this is even more so in mutants of the MLL compass complex. They are shorter lived, while there's a significant lifespan extension upon UV treatment of the SBR5 demethylase mutants. Then we directly interrogated for the dimethylation pattern, and indeed at 24 hours, we saw a transient increase in the dimethylation, but no changes in tri and monomethylation, suggesting that there's a transient and dynamic regulation. And interestingly, 24 hours is the time when the lesions that are induced by UV have been already repaired. And we see that in situ as well. At the same time, there's a strong increase in dimethylation, quantified here. But another modification is control is not out. Then we hypothesized that maybe the dimethylation could have something to do with gene expression. And here at 24 hours, we look at genes that are differentially expressed and that have a differential deposition of dimethylation, and we see a strong enrichment of induced genes at the same time point. We look further at these genes and we find that the processes they're involved in are ranging from RNA transport, spliceosome, ribosome biogenesis, translation, mRNA surveillance, protein processing, proteasome, everything kind of connected to protein biosynthesis and homeostasis. So then we hypothesized that it is known that after UV, at a time point 6 hours where lesions are still present while they are being removed, it is known that many genes are downregulated, but they need to recover after lesion repair. So here we look at the genes that are down at 6 and recovered, upregulated by 24, and a very large number of those has indeed an increase of dimethylation. And again, we see protein processing, proteasome, and so on. So then we hypothesized that maybe this could have to do with protein biosynthesis. We used heavy lysine incorporation to measure protein biosynthesis. We see at 6 hours, when lesions are still present, we see that there's a strong reduction, just about over 30% of the non-treated control incorporation of the heavy lysine. And this gradually recovers in a wild-type animal, and it more profoundly recovers in mutants of the demethylase SBR5, while the MLL compass component WDR5, those mutants fail to recover, suggesting that this has really consequences. Now we look more specifically here at proteins of the spliceosome and proteasome. What we find is that there is a gradual increase over time in the abundance of these proteins, in the translation of these proteins, much more pronounced so in the SBR5 mutant, but it's flat when the MLL compass component is defective. And we see this is reflected in the recovery of the genes that underlie these proteins, where there's a recovery in wild type and SBR5, but the recovery fails in the MLL compass mutant complex. So it's particularly striking in ribosomal proteins because there's really a complete loss of their de novo translation in the MLL compass component mutant here, while they recover very effectively in the demethylase mutant. And this is reflected by the expression of some of these genes that we investigated. Now, this led us to the hypothesis that maybe protein biosynthesis is required for the animals to become resistant and recover and maintain development and lifespan after DNA damage. And so here we use very low doses of cyclohexamide, a very well-established protein synthesis inhibitor that on its own, in the absence of UV, at this low concentration has no adverse effects. But then after UV treatment, there's a strongly exacerbated UV sensitivity, and the resistance that the SBR5 mutant confers here, so they develop further than wild type, this is completely abrogated by the presence of cyclohexamide, indicating that the augmented recovery of protein biosynthesis underlies the resistance of these animals. And this is true also for their lifespan. So here again, on UV treatment in solid lines, there is reduction of lifespan, still an extension of SBR5, they are resistant, and this resistance is abrogated when protein biosynthesis is abolished. So our model here is that UV-induced DNA lesions, they lead to an inhibition of transcription activity. Consequently, also protein translation is stored. These lesions are repaired by transcription-coupled nucleotide acid repair, and upon their repair, the MLL compass complex deposits dimethylation on gene bodies of genes that are involved in protein biosynthesis and homeostasis. They are then transcribed again, and they are promoting through recovery of protein biosynthesis, developmental growth, as well as maintaining longevity upon DNA damage. So to summarize this part, upon UVD-induced DNA damage, his H3K4 dimethylation is deposited by the amino complex complex, removed by the SPR5 demethylase. This dimethylation is essential for resuming developmental growth and lifespan after DNA damage. The DNA damage-induced dimethylation increases triggered by the completion of repair data. I didn't have time to show you. This increased dimethylation is associated with transcription activation and protein synthesis and homeostasis genes. And the dimethylation regulates post-repair recovery of protein biosynthesis and homeostasis, which is essential for maintaining developmental growth and lifespan amid toxic stress. In the last 2 minutes, I just want to switch gears and tell you a little bit about aging biomarker development that we have recently been interested in, also based actually on C. elegans work, because as you all know, this is really a prerequisite for anti-aging medicine. We've heard a lot about aging clocks, the very precise methylation clocks. However, they are not present in all organisms, for example, not in C. elegans, and there's also different effects on cellular composition. There are proteome clocks, which also sometimes have very high accuracy, but the standardization can be challenging depending on the different labs. Transcriptomic clocks have been pursued for quite some time. The advantage is that it provides information on the underlying genes, but the accuracy is limited by variation in gene expression levels, which can be considerably high. Now, we have started this from C. elegans, where we had almost 1,000 C. elegans RNAs sequencing samples from all type of longevity intervention to derive a transcriptome clock, and that's what David Meyer has done in the lab. What David did was he used temporal scaling, which in C. elegans, of course, works very well because the age, the lifespan of all these strains, for example, is known. And he then developed a binarization approach, which really reduced the variability and gave us an extraordinary high indication of aging based on these transcriptome data. And this indeed is very close to the theoretical limit of a prediction of a biological age clock. Now, we tested many interventions that are known in C. elegans. DAF2, for example, already early in life predicts in nearly twofold extension of lifespan. Dietary restriction is predicted. Also, the prediction of one of the strongest lifespan extension mutants, DAF2-RSK1. Also, conversely, lifespan reduction is early predicted in life, here, for example, by a microRNA mutant. We also predict that the gain of function SKIN1 accelerates aging, which is suppressed by WDR5, and also a disturbed proteostasis that accelerates aging can be accurately predicted. Now, this looks now like a lot of graphs, but just to make the point, dietary restriction the lifespan effect can be predicted, but not in a mutant that is refractory to this. The lifespan shortening of heat stress, as well as of pathogenic bacteria infection, here 2 distinct ones can be predicted. Also a difference in a metabolite that actually only shows at 12 days an effect on health span can be accurately predicted at the late life, but not in early life. Also, the metformin treatment on bacteria that allow the lifespan extension on metformin, but not the resistant bacteria, where metformin does not extend C. elegans lifespan. Also, treatments with here an antidepressant that extends lifespan can be predicted. A triple combination of a drug treatment that has double combination here it had no effect in the lifespan extension, and also disturbed proteasomal function by an inhibitor that at high doses has the same effect as a mutation in a proteasomal component. Now, because they are genes, we can here derive the processes, and we find, interestingly, the genes that underlie this aging clock are mostly genes with a signal peptide that are associated to an innate immune response, which is secreted and probably systemic, which might be a driving factor of the aging process here. This also works in humans. Here we take human fibroblast from all different ages. With a binarization approach, we find a very accurate prediction, as well as the lifespan shortening, the accelerated aging of Hutchins-Gilford progeria fibroblasts. This binarization also improves aging clocks in human blood samples, where previously the state of the art has been rather low accuracy, which can be strongly improved by our binarization approaches to a very high Pearson correlation, for example. So essentially, we validated this elegans clock and also in human whole blood samples. It's a simple assessment of biological age, which is applicable to all model organisms research projects. I thank the people who did the work, particularly C.R. Wang, who has really done the project on the amino compass complex. And I thank you very much for your attention. If any of you is more interested, we do have a postdoc position available if you're interested in this line of work. And I thank you again, and I'll be happy to take questions.
So thank you, Bjorn, amazing lecture, and covered a lot of ground, including the biomarkers. Read the paper, recommended it when it came online on archive. There are many questions from the audience, so I'll start with Wilbert Vermeij. I hope I'm pronouncing it correctly. So when comparing the lifespans of non-UV treated worms, was there any effect on CHX treatment itself?
So yes, we do see a lifespan extension of cyclohexamide treatment in the absence of UV. So yes, this is really interesting, and Wilbert picked this up really well, that actually under normal conditions, inhibition of translation extends lifespan. I mean, that's how TOR signaling adaptation extends lifespan. So under tonic condition, this does promote lifespan extension. However, for acute treatments, as we induce it by DNA damage, it's required for this organism to actually boost protein biosynthesis. And I think this is really fascinating because it suggests that under different conditions, the promotion or inhibition of protein biosynthesis can have very distinct consequences. Sometimes it's required for the organisms under acute stress, and tonically over a very long time span, you need to reduce it actually.
And one last question, so by the co-chair, Morten, have you looked at the SAM levels after DNA damage?
The SAM levels, no, we haven't looked at them.
Got it. So thank you very much. Again, to everybody in the audience, please look up Dr. Bjorn Schumacher's book, Mystery of Human Aging, join his lab as a postdoc, and add him on Twitter, Schumacher BG. That's the Twitter handle. It's always good to be informed. And we are